Well, good afternoon, everyone, and welcome to our program, Impact of Aging and Exercise on Immunity. I am Vicki Newell, and I manage the volunteer program. I think you first saw me with a mask on. We do wear masks around here, but in this large room, we don't need them because we can maintain 6-foot distance. And so you can understand us a little better. Marian, the presenter, and I will not be wearing our masks. Just a reminder to submit a question in the upper right-hand corner of your screen. You should see a red box with a white arrow in it. If you click on that, it will expand. And it'll look something like this box. And at the bottom, there'll be enter a question for staff. So enter your question there and just hit submit, and we will get to those at the end of our presentation. Our presenter today is Marian Kohut. Marian received her bachelor's degree in biology from the University of Chicago, her master's degree in physiology from Penn State University, her PhD in exercise physiology from the University of South Carolina, and she completed her postdoctoral training in psychoneuroimmunology from the University of Rochester in New York. Been all across the country. She has been at Iowa State University for 20 years and is currently a full professor and is the Barbara Forker Endowed Professor in the Department of Kinesiology. She is a faculty member in graduate programs of Immunobiology, Gerontology, and Neuroscience, and she is a member of the Nanovaccine Institute. Please welcome Dr. Kohut.
All right, well, thank you very much, and happy to be here today to share a little bit about some of our research, aging and immunity in general. So I'll first start with describing the field of geroscience. Geroscience is really a newer way to approach aging research. If you know, most of the medical research in the United States is funded by the National Institutes of Health. And the tradition in that NIH has been to focus on specific diseases. So for example, there's the National Heart, Lung, and Blood Institute, so they focus on heart disease, for example. There's a specific focus on Alzheimer's, and so on. But there was a shift in this kind of thinking that really took place around 2012, 2013, 2014. And based on research that has been done in aging biology, we have learned that many of the age-associated deficits have some common mechanisms. And research with model organisms has shown that therapy strategies that can prolong lifespan in different model organisms also improve what we call health span. So we see reduced incidence of chronic type diseases or delayed onset of those types of diseases. And so there's been this shift in thinking that rather than focusing on specific diseases, if we could better understand aging biology, that may be able to impact multiple diseases. And that's really sort of the new path forward in developing treatments for multiple types of age-related diseases. We also recognize that aging itself is a risk factor for many diseases. And so getting a better understanding, developing an understanding of these age-related biological mechanisms may have implications, again, for multiple diseases. So this has taken off. The Geroscience interest group at NIH, they had another meeting in 2017, and it's a group of researchers in the field who have worked together to progress this line of research. So as a result of this initial Geroscience Summit, what they did at this initial meeting was to look at the field of aging biology and try to define mechanisms that we see that underlie some of the age-related changes. And these have been broken then into different categories. As a result of that first summit, 7 pillars of aging and geroscience were put forward, which are seen here. These include macromolecular damage, such as damage to, say, mitochondrial cells, epigenetics, so changes in that impact gene expression, inflammation. We see increased inflammation with aging, adaptations to stress, and these can be multiple types of stressors, ranging from cellular stress all the way up to psychological stress that may impact aging systems. Proteostasis, so managing protein health, stem cells and regeneration, and then metabolism, another area in which we see age-related changes. So one other item to point out here is there's really not one mechanism per se that underlies aging biology, but rather not only do all of these factors contribute, but there is a high degree of connectedness shown by these lines connecting these, such that if you impact one of them, that may have impact on several of the others. So it's a complex picture. Here is another similar type of graphic, again showing these different biological processes of aging. And essentially, you may see these organized in different ways, different places, but most of the time they're grouped into between 5 and 10 different types of biological processes. You can group them slightly different ways, but the biological processes remain essentially the same. So just kind of showing you another image here. And then the question, especially for biomedical researchers, is how can we use this field of geroscience to address age-related conditions? So as you see here on the left are our list of some of these mechanisms, these biological mechanisms associated with aging. And NIH has invested quite a bit of effort in looking at different interventions, some of these you may have heard of, that can impact some of these basic biological aging processes. And the idea here is that some of these different interventions may impact multiple processes and then in turn may have a benefit for multiple types of disease, for example, cardiovascular or osteoarthritis. Also, these types of interventions may influence what we consider geriatric syndromes, such as frailty, delirium, cognitive decline, and so on, and also even have an effect on resilience, the ability to withstand insults to the body that occur with aging, for example, hospitalization, infection, and so on. So again, the approach here is to really better understand the biological mechanisms of aging, target or identify interventions that might target multiple of these processes, which then can have benefits across multiple age-related conditions instead of looking at individual conditions, diseases by themselves. So this is really the newer approach to aging. And I just added this slide here. You might find this interesting. There's a link to the NIH website that describes what this interventions testing program, which was started fairly recently. Right now, the interventions are being tested in mice as the model system. And this includes really a wide range of different approaches, from pharmaceuticals, nutraceuticals, different foods, diets, dietary supplements, hormones, peptides, and so on. So there's quite a long list. If you find this interesting, you might want to go to the website. There are already some publications from some of these that have been tested, and quite a few of them show early promise. And again, we're beginning in mice, although some of these have already been tested in humans. But the idea is to try to define interventions that are effective across multiple mechanisms of aging. So in my research lab, we have had a focus on physical exercise and the aging process. And one of the reasons for this is that exercise has been shown to impact macromolecular damage, epigenetics, inflammation processes. We have some early work that we did quite a few years ago, and now there's quite a bit of evidence showing that exercise can reduce inflammation levels, adaptation to stress, proteostasis, stem cell, and regeneration. and metabolism. And so our approach has been to use exercise rather than, say, a pharmaceutical agent to target these different biological processes that underlie aging as a means to attenuate age-related diseases and conditions. So in our lab in particular, we've been focused on two areas. One of these is the area of immunosenescence. With advancing age, there is a remodeling of the immune system that takes place that is often termed immunosenescence. Features of immunosenescence include an increased susceptibility to infectious disease as we age, reduced response to vaccination, for example, increased susceptibility in particular to age-related inflammatory diseases. Also, as part of the aging process, as we mentioned, one of the biological features we see is inflammation. And this is really a chronic, persistent, low-level degree of inflammation, and it's been termed inflammaging. What we know is that this higher level of inflammation might impair optimal immune response, so the body cannot elicit as productive an immune response as when one was younger. We also know that this chronic low-level inflammation is associated with multiple chronic diseases, including, for example, diabetes, heart disease, Alzheimer's, other neurodegenerative diseases, obesity, and so on. So even in younger adults, that may be affected with diabetes, for example, we tend to see higher levels of chronic inflammation. So those have been two areas in which we have focused in particular. And today I'm going to share with you several of the research studies in our laboratory. We started looking at exercise and immune response to influenza vaccine in older adults. Then we also included in our research the impact of exercise on inflammation, this chronic low-level inflammation that we see, and also have moved to a model in which we can study respiratory viral infection, as we know that oftentimes morbidity and mortality from respiratory infections is increased with aging. So I'll share with you, this is really quite an old slide now. I mean, you usually don't even see these colors in the slides, but I saved my old slide because this shows where our work first started, actually when I first arrived at Iowa State University. And one of the questions we had at that time was, can exercise have an impact on immune function in older adults? One way to study this is to look at the response to flu vaccine. Most older adults are immunized regularly, so it's a nice model to study the immune response. And we also have a clear immune outcome that we are looking for in terms of an optimal antibody response. Higher antibody response suggests greater protection from infection. So with our very first study, we went out to the community to find individuals who were planning to be vaccinated, and we simply asked them how much exercise do they typically do. So we grouped people into three groups: sedentary, those were older adults who did not participate in any regular exercise. They were still, however, capable of activities of daily living, didn't have limitations there. Then we had a group which we defined as moderately active. These individuals exercised less than 3 times a week at a higher intensity level, but they regularly did perform some type of lower intensity exercise, for example, walking. And then we had what we called our most active group. This group exercised at least 3 times a week at what we call a vigorous level. So that's sufficient to raise your heart rate and breathing. That could be very brisk walking. For some people, it might be slow jogging, cycling, something like that. And so then, after people were immunized, we asked them to come to the lab, give a blood sample, and we really looked at antibody response. Here we just have different dilutions of serum. More concentrated serum, we'll see a little more antibody. But what the pattern is that those who are most active had the highest antibody response to flu vaccine. And there really was no difference, although it might look that moderately active was less than sedentary. Statistically, these were not different. But so this is some of our first evidence that exercise might have some benefit on immune response. However, that type of study, you could always make the argument that those who were the healthiest were the ones who were most active. And so we went on to do a randomized trial in which here we started with all older adults who were currently sedentary. And then they were randomized into one of two groups. One is to continue their current daily habits, not really change anything, so they didn't exercise regularly, or they were assigned to begin an aerobic exercise program, which they did for approximately just a little bit less than a year, 10 months, 3 days a week, 30 to 45 minutes per day. And this was exercise that was aerobic primarily in nature. So we had participants come in, walk on the treadmill, for example, or bicycle, and they did that again for roughly 10 months. So with the study design, people were immunized 1 year. We measured their antibody response to the flu vaccine, and then they started either their exercise or control treatment, which they did until really the next flu season, in which they were immunized again, and we measured antibody 1 month after the flu vaccine. And so here I do want to point out that we actually did include young adults as a control comparison group. And what we're looking at here on this slide, the top graph is the antibody response to influenza A, H1N1. Also, that vaccine contains influenza B or influenza A, H3N2. So we measured all of those. I'm showing the data for H1N1 and influenza B. It was really the same pattern across all strains of flu. What we're looking at here is 4 weeks post-immunization or 3 months post-immunization. The black bar are older adults who maintain their sedentary habits for the year. The white bar are older adults who began an exercise program, and the red bar are 20-something-year-old college students. So what we see here at 4 weeks post-immunization, older adults do increase. We're comparing this to pre-immunization levels, so anything above 0 is an increase. As you can see, older adults who exercised had a better antibody response than those who did not. Exercise didn't quite get us back to being 20 years old again. However, I think we'd rather be in this group than this group. One interesting finding we saw with this study is that by 3 months post-immunization, our older adults who didn't exercise, their antibody levels dropped back below pre-immunization levels. So this would suggest not very good protection at this time point. Usually that declines slowly over time. This particular group, we did see a big drop in that. But again, the pattern is the same, that exercise is better than no exercise, not quite as good as young adults. And the same pattern here for influenza B. So this suggested then that, again, more evidence that exercise can improve antibody response to the vaccine. So then we went on to take this a little bit further and try to address what might be some of the mechanisms by which exercise can have benefits. We do know that, again, adaptations to stress is one of the important mechanisms that can underlie biological aging. This does include psychosocial factors as well. So in this next study, we compared different types of exercise. We also measured psychosocial factors such as stress, depression, and so on, as one possible way in which exercise might have benefits on immune response. The other area in which we looked a little bit deeper, we know that the neuroendocrine system, hormones, for example, interact with the immune system, and the immune system communicates with the neuroendocrine system. When you exercise, there are changes in these neuroendocrine factors that could influence immune response. So we also did a little study to look at a couple of these neuroendocrine factors, specifically epinephrine and norepinephrine, which I'll explain in a minute. Okay, so our study design here, Our question was, how might exercise, again, alter immunity to vaccine? And can exercise help reduce inflammation, this inflammaging? So we have exercise over here. We do know that exercise can help improve psychosocial factors such as reducing stress and so on. We also know that high stress levels, for example, caregiving stress in older adults, can be associated with reduced vaccine responses. And so by reducing or by improving these psychosocial factors, that might be one pathway by which we see improved immune response and less of this immunosenescence. Then to address the neuroendocrine factors, to see if those might impact inflammaging, we did include older adults in the study who were taking a class of medications called beta blockers. Those impact modulate the effect of epinephrine and norepinephrine on immune cells. And so we looked to see if the response was any different in older adults who were taking that group of medications. And so here is our study design. A few more arrows here, but I'll walk you through this. Again, at the beginning of the study, everybody was vaccinated, and we measured their antibody response to the vaccine 4 weeks later. After that, they began either an aerobic exercise intervention for 10 months or a flexibility strength training type of program in which there was some walking as a warm-up, but then individuals focused more on toning types of exercises, stretching, as well as some weightlifting. Individuals completed psychosocial surveys at the beginning and then again after the 10 months of exercise treatment. Again, people were immunized for the next year's flu vaccine. We measured antibody response before, immunized 4 weeks and 12 weeks later. And so in terms of responses that we are measuring as far as vaccine response, B cells make antibody. As I mentioned, higher levels of antibodies is correlated with better protection against infection. Although the current flu vaccine is targeted primarily towards inducing antibody, there can be some T cell immune responses as well. And some data suggests that T cell immunity may be better correlated with protection from infection in older adults than antibody level. So that is why we measured T cell responses as well. Just very briefly, the T cells have an important role in killing virally infected cells. So for example, let's say there was a poor mismatch in a given year between the vaccine antigen circulating and what was in the vaccine, and someone was to become infected, that T cell immunity might help provide some protection. So another reason for looking at T cell response. And then here's just a sample of blood in which we also measured these inflammatory biomarkers. Okay, so our findings, not too surprising on the psychosocial side. Here I'm showing results from depression, stress measure, sense of coherence, which is really a measure of sort of purpose in life, so a positive kind of resilience factor, or optimism. And then we see the flexibility strength group and the cardio exercise group. We can see that both groups had a drop in depression scores, so that's positive. The stress score actually declined in the cardio group, but it did not change in the flexibility strength group. Sense of coherence and optimism improved in both groups. So in a way, this was not too surprising, as other data suggests that exercise can have positive psychosocial benefits. But it was nice to see this in older adults. However, in terms of this pathway, and I won't get into the statistical modeling here, but I have help from some statisticians who can do more complex modeling to determine, are these effects from exercise on immune response mediated by these improvements in psychosocial factors? And the short answer to that is no, they were not. While exercise did improve psychosocial factors and did improve antibody response and T cell response, it was not through the change in psychosocial factors. And we'll come back to neuroendocrine in just a moment. So one item that I wanted to point out that I haven't mentioned yet is that obesity can also be associated with poorer response to vaccines. So after the initial vaccine, the first vaccine before they started exercising, we did look at antibody response, comparing that in obese. Those are individuals with the body mass index over 30 or non-obese. Here on this axis, we see, again, the antibody response to H1N1 at 4 weeks or 12 weeks, the antibody response here, in this case, to H3N2 flu virus. And you can see the pattern here really across time points and across viruses is that those who are non-obese have a better immune antibody response to the vaccine than those who are obese. So that gave us a clue that we may have to consider this in our findings. So the data on this slide shows really 4 different groups, and I'll explain this briefly here. What we found essentially was that cardio type of exercises versus flexibility strength exercises, one was not superior to the other in terms of improving immune response to the vaccine. But what we did find was important is that improvement in fitness level from the start to 10 months later was important in determining how well somebody responded to the vaccine. So it really didn't matter if you were doing cardiovascular exercise or flexibility strength. Instead, how much you improved in fitness level, and we measured that with a walk test or treadmill test, how much the degree of improvement there was more important than, again, what type of exercise you did. So here again, we see antibody response, again, to H1N1 or to influenza B. And we have 4 groups here. Those who improved in fitness, again, either type of exercise, and were non-obese are in the black bar. Those who did not change in their fitness level significantly, but were non-obese. Then we have in the green color, those who improved fitness level and were obese, or the yellow, those who did not improve fitness significantly and were obese. And you can see a pattern here across time and, again, vaccine strain, in that those who improved fitness, so that would be our green and our black bars, tended to have a higher antibody response to the vaccine than those who did not improve fitness, regardless of whether they were obese or non-obese. And that pattern held consistent, again, across time points. So in this slide, we're looking at the T cell response to the vaccine, and we see really that same type of pattern. So that those who improved fitness, again, the green or the black bars, had a higher T cell response, again, to the 3 different antigens in the flu, H1N1, H3N2, or type B influenza. And furthermore here, I just wanted to point out that if we're comparing just the obese individuals, those who improved fitness compared to those who did not, between these 2 groups, there actually was a significant difference as well, in that improving fitness, even if one was obese, definitely had a higher antibody response, or sorry, not antibody response, T cell response, than those who were obese and did not improve fitness. So quick summary here. Again, overall improvement in aerobic fitness level was more important than specific type of exercise. And again, we saw that non-obese tended to have a better response than obese, but fitness was certainly a factor here. And T-cell response, again, was greater in subjects that improved fitness, again, regardless of what type of exercise training they did. Then in terms of looking at inflammatory responses, how might exercise impact inflammaging? Again, we looked at the type of exercise and the role of these neuroendocrine factors. And I'll just briefly show you data here. Interleukin-6, this is an inflammatory protein. It has, in fact, been correlated with morbidity, mortality in older adults. Higher levels of IL-6 are associated with greater morbidity and, in fact, greater rates of mortality. So we have our 4 groups here, but in this case, fitness level, the change in fitness level seemed to be less important than the type of exercise. So we are comparing cardio exercise in the orange circle, flexibility exercise in that dark red circle. Flexibility exercise is a light green circle in people who are taking beta blockers, again, that those class of drugs that can block the effects of epinephrine or norepinephrine on immune cells to some extent, or those who were in the cardio group taking beta blockers. The pattern here we see for IL-6 is that those who are in the cardio group, regardless of whether they're on beta blockers or not, had a decrease in IL-6 from the beginning of the study to the end of the exercise intervention. And that's what we were looking for, a decrease in inflammation. In contrast, those who did the flexibility exercise that were not on beta blockers had just a very modest decrease, and those who were taking beta blockers actually had an increase. If we look at another inflammatory protein, IL-18, we see the similar pattern here in that those who were in the cardio group had a bigger drop in IL-18, and those who were in the flexibility strength group really did not have much of a change. C-reactive protein, CRP for short, was also measured. We see a similar pattern in that cardio tended to decrease the C-reactive protein levels to a slightly greater level than flexibility strength group, and we actually saw a slight increase in the CRP level in those who did the flexibility exercise. So here the pattern was a little bit different in that the type of exercise was more important. We saw that the reduction in inflammation tended to be greater in those who participated in cardiovascular exercise rather than strength flexibility exercise. And although I'm not showing the data here, we also looked at the role of psychosocial factors, body fat, because that can impact inflammation. And again, the beta blockers, we saw a little bit of that. But basically, the effect of exercise was independent of changes in body fat, psychosocial factors, or catecholamines. Okay, so now we're going to move to the mouse model, and primarily for ethical reasons. In terms of studying respiratory response to influenza, infecting older adults with influenza has, of course, ethical concerns, so we use a mouse model, and we use aged mice. And so the question here is, now moving from vaccine to actual infection, does exercise have any benefits in terms of respiratory viral infection? Can there be any protection from exercise? So we use a mouse, and because we've been studying influenza with our vaccine, and also, this is before COVID, we know that the morbidity and mortality from influenza is much greater in adults who are over age 65, similar to what we see with COVID. So in these studies, we have aged mice. They were assigned to an exercise group in which they run on a little mouse treadmill 5 times a week, 45 minutes a session. Is kind of a moderate intensity for 3 months or more. The control mice get exposed to the treadmill and the stress of handling, but they don't actually run. And after this training period, mice are all rested for at least 1 day, and then we'll infect them. We'll drop flu virus into their noses and see how well they respond. And this is just a picture of mice actually running on the treadmill in our lab. Mice, unlike rats, really do like to run on the treadmill. Rats, oftentimes you have to shock them to get them to run, and that induces a lot more stress. But mice are good little runners, especially if we make it dark towards the front of the treadmill. They'll run up to the front of the treadmill happily for quite a long time. And here's one of our former students, who's now actually a physician, but she helped do these studies quite a long time ago with our aged mice. So in a mouse model of flu infection, what we see, which is similar to some extent in humans, is there's weight loss. And the degree of weight loss is correlated with the severity of infection. So if we look at the graph on the right here, we have exercise mice in the red bar and in the red line, and non-exercise mice in the black. After they're infected, we have days here, day 1, 2, 3, all the way up to day 9. We monitor their body weight. And as you can see that aged mice who've been exercise training have less weight loss during flu infection than mice who did not exercise. When we look at recovery from infection, usually around day 8 to 9, there's an inflection point, and they start to turn the corner from the infection and regain weight. As you can see, this is, again, another group of mice. Here, the exercise are in white instead of red. Apologize for the change in colors here. But as you can see, the exercise mice around that day 8 to 9, they start bouncing back more quickly and regain weight compared to non-exercise mice. And we've seen this pattern across multiple experiments. Exercisers regain their health more quickly, and we have less morbidity. And if we use a high dose that induces mortality, we see that exercise mice have better protection from mortality than non-exercise mice. Okay, this next slide has a lot of graphs on it, but I just want to point out a few key points here. One is we did measure how much virus was actually in the lungs of mice. And one concern with exercise is that there might be changes in the lung structural changes so that perhaps exercise can prevent less virus from getting into the lungs. Typically, when we put virus in, we see it start to replicate within 6 to 8 hours. By 24 hours, you have a big jump in virus in the lungs that continues to increase to about day 3 or 4. So what we did here is in one study, we actually inactivated the virus, and so it could not replicate, but instilled it into the mice to see is there a different amount of virus that gets into the lungs in exercise compared to non-exercise. And as you can see here, at either 6 or 24 hours, there's no difference. So any effect on viral load is not related to maybe some type of structural changes that allows a different amount of virus to get into the lungs. So then we look at different time points and different doses of virus. What we can do in the lungs is what's called bronchoalveolar lavage. That's a wash of the lungs, and that collects fluid that would be found on the surface of the epithelial cells, in which influenza virus infects those epithelial cells. So some virus is found there, and some gets then deeper into the lung tissue. What we see at either 12 hours post-infection or 24 hours, the pattern here is that our non-exercisers are in the black, exercisers are in the gray. There is a modest decline in the amount of virus in the BAL fluid, we call it, or in the lungs. If we use a low dose of virus, by day 4 post-infection, that's usually our peak viral load, we see that there's a significant decrease in the amount of virus in the BAL fluid of exercise mice. At day 4, we didn't see a difference when we used a high dose of virus in the BAL fluid, but in terms of how much is getting into the deeper layers in the lungs with the high dose of virus, again, that was reduced by exercise. If we look later in infection, these are viral load measures. There doesn't seem to be much of a difference between the amount of virus found in the lungs, and by that time point at day 8 or day 10, that virus is being killed and cleared by the immune system. So we don't see a difference in load, but if we look at the percent of mice that actually have virus left in their lungs at that time point, the percentage of mice, we can see that at day 8, we have just roughly less than 20% of the exercise mice with virus still present in their lungs, whereas almost double, 40% of the non-exercise mice, and a similar pattern with a high dose of virus at day 10. So exercise seems to reduce that early viral load, speeds up the clearance of virus, and again, these effects seem to be immune-related rather than mechanical per se. Now, this slide has a lot of figures on it. I won't go into detail on all of them, but if you'd like to look at them later, you can. But what we are measuring here are chemicals called chemokines. These recruit immune cells to the lungs. Now, we do want to recruit immune cells to the lungs to initiate an immune response, but an over-exaggeration of immune cells, an influx into the lungs, could actually be potentially damaging. So there's a balance that we're looking for here. Here I'm showing data from non-infected mice are in the black bar, which you can barely see on most of these graphs, and that's because non-infected, we shouldn't have a lot of these chemicals expressed. Infected, non-exercised are the white bars, and infected mice exercised are the light gray. The pattern you pretty much see across almost all of these chemokines at either day 4, which is the first point on each graph, or day 7, is that the exercised mice in the gray have fewer of these chemokines than non-exercised mice. And so again, that pattern is pretty consistent across time points and different chemokines. This is a similar type of slide. Instead of looking at chemokines, we are looking at what's called cytokines. Those are proteins that are involved in immune activation, inflammation, and we see that same kind of pattern here, that the non-exercised mice in the white bars tend to have more of these inflammatory mediators than exercised mice. So a little bit less inflammation in the lungs. Again, that's at day 4 and at day 7. This, not surprisingly then, is followed by immune cells being recruited to the lungs. We look at different populations. Those that are recruited to the lungs with infection include inflammatory monocytes, neutrophils, dendritic cells. The alveolar macrophages, those are largely resident, already present in the lungs. And so we can see, again, that same type of pattern in that non-exercised mice have more of these immune cells being recruited to the lungs than exercised mice. So at first, that was a little bit challenging to interpret because we were thinking that, well, with exercise will enhance immune response. And so many of these measures that we're looking at early on, we see actually that there is less in exercise mice. So we did look at some later time points to see if we see a different picture. On the right here, we're looking at T cell response. If you remember, I mentioned that T cells are important in killing virus-infected cells. And T cell responses are directed against different influenza proteins. We see a couple different proteins in this graph. What we're measuring here are the number of T cells. Oops, this is moving around a little bit. There we go. Number of T cells in the lungs, and then the effectiveness of the T cells in terms of producing a chemical that helps in lysing virally infected cells. So here we see that, again, non-infected mice, we don't expect to see as much of a specific T cell response as infected mice. Exercised mice are the gray bar, non-exercised in the white. Here we really don't see a difference in the number of T cells responding to these flu proteins. But on the right side, when we look at the T cells that are producing interferon gamma, those are reflective of T cells that are involved in killing virus-infected cells. We can see that the exercised mice, again, tend to have higher T cell response than non-exercised mice. And I'm not showing all the histopathology here, but we have a pathologist who looked at the lungs of these mice. And the short story is that exercised mice later in infection have less damage in the lungs than non-exercised mice, which would be consistent with a little bit less inflammation. So as I mentioned a moment ago, well, we do see some benefit on the T cell side that might help explain why mice do better. But we were still questioning why even early on in infection, by day 1, by day 2, we see less virus in the lungs of exercised mice. And so we started to focus on some of the very early antiviral responses to infection. And the type 1 interferons are a group of proteins that can interfere with viral replication. In fact, that's how they were named. So you really can't see from this graph, so I apologize that it's not great for viewing. But briefly, what we're looking at is different time points, 6 hours, 12 hours, 24 hours, so very early on in infection. We know that these interferons can induce a series of genes to be expressed that can help limit viral replication. And so I know you can't tell from these figures here, but basically what this showed us is that at the very early time points, 6 hours for sure, and 12 hours to some extent, we are starting to see more of these interferon-induced genes expressed in the lungs of exercise mice compared to non-exercise mice. So some benefit very early on gave us a clue that interferon might be a protein that's upregulated with exercise training. And the reason I'm bringing this up, it has relevance in other disease models as well. This is actually some work from humans in which we collected immune cells, infected those cells with virus, and we looked at how much of these inflammatory proteins relative to interferons are produced. And this is data not from older adults, but from middle-aged adults in which some were normal and some were pre-diabetic. And we see a similar skewing to what we see with aging in that the ratio of inflammatory cytokine to interferon is higher in those with prediabetes compared to normal adults. This is a similar pattern to what we've seen in aging and true of another inflammatory protein. So again, in diseases associated with inflammation, which includes aging as a condition or prediabetes, we see this shift towards an inflammatory rather than an antiviral interferon response. And as again, I mentioned this because this may have implications for other pathogens that interferon plays a role in limiting viral replication, such as SARS-CoV-2, so COVID-19. So then, just to get a little bit deeper into this question, what we did is we looked at, we treated mice with anti-interferon antibody, which basically blocks the effect of interferon. And what we see here, what we can study here is the role of interferon in providing protection in our exercise mice. So basically, we have mice treated with an antibody that blocks interferon. Some of those mice exercised, some were control, or mice that were not treated with antibody. Again, exercise or control mice. If interferon is a mechanism by which exercise provides protection, then the mice treated with antibody, exercise mice treated with antibody, won't have that benefit of exercise. And that's actually what we see here, is that exercise mice, no antibody treatment, again have less weight loss with flu infection. But if the exercise mice are given this treatment to block interferon, they are just like our control mice. So that suggests that this early production of interferon might be an important pathway by which exercise can provide some protection from infection. And then here is really just a summary of what we saw in the slides. And I just have a couple more slides here. I don't want to run out of time for questions. So I'll move on just to let you know about some of our ongoing work. We have several projects going on right now. One of these is focused on immunity and aging. And here we are really trying to get at which of these underlying biological mechanisms contribute to immunosenescence. We are also asking how might lifestyle factors, psychosocial factors, for example, impact these and then ultimately impact immunosenescence. We also have a group of people in our research, on our research team that are looking at cognition and the impact, again, of these different biological pathways. Then on this side, we're collaborating with others who are working to develop interventions or different strategies that can target these pathways, hopefully then reducing some of the negative effects. So I just put a slide up here. This is our Immunity and Aging project. We are recruiting participants. Right now, older adults 65 and older participation is on hold, but with vaccines rolling out, we expect that'll change soon. Then the second area is nanovaccines. We just recently received a grant that is focused on optimizing nanoparticle-based vaccines for older adults. So these vaccines are targeting older adults specifically and will be designed differently than the current influenza vaccines. And that's a study for which we are also recruiting participants, as we'll collect blood from older adults as well as mice and test these components of the nanovaccine. So a little blurb about that. And then our last one, I'll just point out, a COVID-19 vaccine study. Here we are looking at a session of exercise just after receiving the vaccine, as some previous work with the flu vaccine showed that people who exercised for 90 minutes, now 90 minutes sounds like a lot, but it was light intensity, brisk walking type of exercise, had a higher antibody response to flu vaccine than people who either did 45 minutes of exercise or no exercise. So we are currently have started a study in which we are looking at the impact of one session of exercise after receiving COVID-19 vaccine to see if that can also boost antibody titer. Right now, that is not recruiting participants over age 65, but if you happen to know someone who might be interested, again, we're recruiting for that study as well. So I will stop here. Thank you for your attention, and I'll be happy to take some questions, and I just put a slide up of all the people that have been involved in this work over the years and recognizing our funding support as well. Thank you very much.
As of right now, we don't have any questions, but I'll kind of do my closing.
Okay.
All right, so as of right now, we don't have any questions, so I'm going to share a little bit of information, and if some pop up, we still have Marian here, and we will have her jump in and answer those for us. So thank you very much. Great program, always very interesting, and I think it's awesome that we have Iowa State University, such a great resource here in town to give us some of this cutting edge information. So thank you very much. Want to remind everyone of our next program that is coming up April 6th. The program is, now I can't find, getting the answers to the questions you have always been afraid to ask. And this is actually about funeral planning. And we have Savannah Pruill, who is with Adams Funeral Home, is giving that program April 6th, 2 p.m., again, a virtual program. We're working on and finalizing our May through August Primetime Alive programs, and those should be out in the next week or two. So be watching for that either via email, or those that we don't have an email for, we will mail those out. And they can always be found on our website, mgmc.org/PTA. There's a section for educational programs. So, and I do not see any questions as of right now. If we get some afterwards, Tim will email them to me, and we'll get those out to Marian, and we'll get those answers to you. So again, thank you very much for the presentation. Thank you for joining us, everyone. Somebody just said, thank you for presenting such a complex topic in a way that was easy to understand. So I couldn't have said it better myself. So thanks.
Happy to take questions.
Marian said she's happy to take questions, so if you send them to me or through this system, we will get those to her. Thank you for joining us, and have a great, I received cortisone shots on 3 to 4 joints. Would T-cell therapy be a better solution?
Okay, the question, I received cortisone shots 3 to 4 joints with T-cell therapy. So, the cortisone shots are to minimize inflammation. T cells are complicated in that they can also contribute to inflammation, And that this is true of inflammation located in different areas throughout the body, different for different conditions. So I don't know of any, quote, T cell therapies per se that are targeted towards reducing inflammation in joints. I know there are a lot of other studies underway to look at anti-inflammatory treatments. Anti-inflammatory treatments such as cortisone has been around for a long time, but there are biologicals that block some of these inflammatory agents, for example, IL-6 or TNF-alpha. Those are used in other chronic inflammatory conditions. To my knowledge, they have not been used locally in a joint, but biologicals as a group are being considered for multiple different types of inflammatory conditions. So, it looks like I have another question. What criteria did you use to determine if exercise was, quote, cardio? Thank you for bringing up that question. I usually mention that, and I think I forgot to define that. Cardiovascular was exercise that was sufficient to get your heart rate up into what we call a moderate intensity, so a intensity zone of about 55 to 75% of maximal what we call oxygen consumption. So when we're not measuring heart rate, if I describe that intensity to people, it's what we would call definitely sufficient enough to get your breathing rate up and your heart rate up. And if you measured heart rate, of course, it depends on age, but probably somewhere between 150 to 160, 170, a little bit lower with advancing age. But so more than a leisurely stroll. Let's see, would I review one more time what studies you are seeking participants for and how do people apply? People can email me, and I don't know if you'll provide email link or how you typically do that.
To do what now?
If they're interested in participating in a study.
Tell them to email me and I'll forward it to you.
Okay. Vicki said to email her and she will forward it to me. Right now at Iowa State, adults over age 65 research has been put on hold due to COVID, but I know they're discussing changing that shortly as that population is largely immunized now. So we are recruiting participants for the Aging Immune General Study. That one is looking at those hallmarks of aging, the biological processes and how they impact immunosenescence, and also how different stress factors like stress in your life, for example, can impact those biological pathways, which then might influence immune response. So we are looking for participants for that, and hopefully it'll be open to over 65 again soon. And then the Nanovaccine study, that is, again, a new study in which we're developing a nanovaccine based on nanoparticles. I collaborate with engineers at Iowa State who designed the nanoparticles, and then the normal flu components, instead of going a typical flu vaccine, are in these nanoparticles, which induces, at least based on our preliminary studies, better immunity. At this point, we are not immunizing people with the new vaccines. We are immunizing mice, but we are collecting blood from humans to isolate immune cells so we can test the vaccine components on the immune cells to make sure they are inducing a better immune response in humans. Because any formulation that moves forward in mice and eventually to humans, we want to have some early data knowing that that's promising for older adults as well. Thank you.
And I'll just one more time.
Okay.
Just one more time. Thank you for joining us, and hope you join us for our next program. And thanks again, Marian. Have a great day.